Hi everyone, my name is Will Echeverria and my presentation will be on imaging genetics and obsessive compulsive disorder. More specifically, on linking genetic variations to alterations in neuroimaging. So many of you may already know or have an idea of what OCD is, but I provided the definition here just as a friendly reminder. So one more time, obsessive compulsive disorder is a heterogeneous psychiatric disorder characterized by clinically significant, recurrent, intrusive, and disturbing thoughts. These are the obsessions, and by repetitive behaviors that are aimed at reducing anxiety or dread, these are the compulsions. Now, the definition that I provided is a more updated version from the article, but the one that's in the slide is from the DSM-5. Now, with this disorder, there's a lifetime prevalence of about 1 to 3% of the population that will be affected. So the article I have chosen decided to focus on a variety of imaging techniques used in past research. With these imaging techniques, it has allowed researchers to look at the brains of OCD patients to look for abnormalities in the neural circuits that control behavior. For most of the past studies, there have been consistent impairments found in the corticostriatothalamic loops. In this region, it regulates neural activity, and these loops are centrally involved in motor, cognitive, and affective processes. They are also assumed to be causally related to OCD symptom generation. In a past study using a structural MRI scan, it revealed that mainly areas of the medial prefrontal wall, which include the anterior cingulate cortex, and adjacent areas such as the orbital frontal cortex, striatum, and the thalamus, were all impaired in OCD patients. In another study, they used diffusion tensor imaging, and it revealed that prefrontal areas like the ACC and OFC that were previously mentioned exhibit altered structural connectivity. These similar regions have also been identified using functional neuroimaging. This has been used during error processing, inhibition, decision-making tasks, and during rest. Even with these past findings and despite the overlapping findings, one should know that these neuroimaging findings in OCD vary across studies. It isn't possible to attribute OCD to a single dysfunctional region or network. So as mentioned earlier when I gave the definition of OCD being a heterogeneous psychiatric disorder, it's because it is a broad spectrum of OCD phenotypes regarding symptomology, severity, and also different genetic backgrounds. OCD has a strong genetic background, but the identification of a single causal gene variance has remained difficult. In many of these molecular genetic studies of OCD, they have investigated candidate genes that are most likely to be at high risk and are significantly associated with OCD. So when it came to looking at the current article, I'll explain a little more about the findings with the serotonergic, dopaminergic, and glutamatergic systems. But these multi-gene risk factors are currently hypothesized to be better predictors for complex disorders such as this. So in the current study, they conducted a systematic review of past research articles, using 63 of them in total. Now, these articles included studies that described imaging genetics in OCD, and a second step included psychiatric disorders known to often be comorbid with OCD, like Tourette's, tic disorder, anxiety, as well as other disorders. So basically, in this present study, they focused on the most significant and nominally significant genes associated with OCD as the inclusion criteria, and they cross-referenced these publications with the first genome-wide association study and meta-analyzed genetic association. I would highly recommend checking out the article. I have it listed in my references, but for time's sake, I'll cut right to what was found and what the researchers in the study were trying to show. The tables provided in the article help with understanding this a little better. So if you get a chance, take a look. So I just want to say that to date, there are only 8 imaging genetic studies that have investigated the association between OCD and genetic variations and that they are published. Most of these genetic associations have been proven by meta-analysis and the Genome-Wide Association Project. But as I mentioned, that's in the article within Table 1. I wasn't able to upload that table because you wouldn't have been able to see it, so when you get a chance, take a look in the article. Now, the researchers found similarities in most of the past research, mainly in the orbital frontal cortex, the anterior cingulate cortex. The striatal and thalamic regions have often been shown to be associated with OCD. So when it came to finding other similarities, there was some involvement with the serotonergic systems. The RAF nuclei are well known to incorporate serotonergic neurons that project to several cortical regions. One of these main target regions that is affected is the orbital frontal cortex. This area is well known for decision-making and learning and is impaired in OCD patients. It must be known, though, that the imaging genetic studies did only focus on the serotonin transporter genes with this finding. It did not include any other serotonergic gene variants. And in the researchers' findings, they stated that there was less known about the impact of dopaminergic gene variations on neuroimaging in OCD. There's only one dopaminergic study that incorporated OCD patients, and they found that the putamen was affected. Now, this part of the brain deals with motor, planning, learning, and execution. All right, so the one finding that was prevalent in most of the researchers' findings was that the glutamatergic neurons are the most prevalent excitatory neurons across the entire cortex, and they play a crucial role in the cortical striatothalamic loops. Now, the glutamatergic genes also influence the thalamus and the ACC. Both regions are assumed to play a role in the cortical striatothalamic loop dysfunction in OCD. Now, the ACC appears to be very consistently involved in OCD, mainly the context of conflict, error, and feedback processing. So with these results, there are some findings that were consistent amongst a good amount of these studies. What this evidence provides is that OCD patients cannot be treated as a homogeneous group in neuroimaging analysis. There is a significant cerebral variability between specific gene variations in OCD patients and controls. Inconsistencies in genetic profiles might also partially explain inconsistencies among neuroimaging studies that focus on OCD. So, this study had a few major limitations. One of them is that there were unequal sample sizes and unequal sized genetic groups. Now, also, neuroimaging is also expensive, so that took a role in this as well. An additional limitation is caused by the pre-selection of genes and brain regions for these studies. And another major limitation was that OCD patients' level of severity differed across the studies, as well as the comorbidities, and this is partially the reason why OCD is just heterogeneous. So, this research helps to show that there's clearly a great need for additional large multi-model imaging genetic studies that assess OCD. Well, that is my presentation. If there are any questions, please feel free to ask on the discussion board. And here are my references. Thank you.